Health is an eternal topic for mankind. We are born on Earth, and our health is intimately linked to the environment and ecology. The environment is the basis for human survival and development, and every subtle change in the environment will eventually have an impact on biodiversity, ecosystem, and our health. Exploring how the environment and ecology affect our health in the past, present, and future is the mission of Eco-Environment and Health. Eco-Environment and Health is a journal initiated by prestigious scientists with a strong editorial board including multidisciplinary scientists from all over the world. With the support from EEH, you will have a better publication experience. EEH has its own offices of publishing, language, design and broadcasting. The experts will accompany you like an harmonious band and spare no effort to sublimate your valuable scientific research into a magnificent and impactful masterpiece. We have a dedicated center for decision making that deeply integrates informatics, ecology and environmental science. It explores global research hotspots, predicts discipline trends, monitors the influence and dissemination of publications, and supports the journal's smart decision-making. Upon receiving your submission, our assistant editors will contact you immediately and keep you informed of the manuscript status throughout the entire process. The 14-day rapid peer review will make the publication of your research one step ahead. Your distinguished work is worthy of being known worldwide. We will promote your research globally in a three-dimensional manner through various social media platforms. In addition, the open access fee will be waived for the first 3 years, making your research easier to be read and cited by your peers. We continue to explore and innovate the service strategy of the journal. EEH leads ecology and environment to a healthy future.
Good morning, everyone. What I'm going to talk about this morning is I'm going to mainly talk about the cultural heritage research. But because I don't want to lead other people, I'm talking about the chemistry and the history, which is the humanity. So I put microbial ecology at the beginning to allow people to understand the background of my information is about microbial ecology. As the organizer has introduced, I have changed my job from previously University of Hong Kong to currently Guangdong Technion, which is a joint university between the one which is in Shantou to the main campus, which is in Haifa of Israel. So in today's briefly talk by me, I'm going to introduce briefly on the civilization, because the civilization related to material and create cultural heritage. So that's really the starting point. And after that, I'm going to give you some summary of our experience working on cultural heritage, particularly in one particular location in Southeast Asia, which is in Cambodia, and the cultural heritage is known as Angkor Monument. And in between and along my presentation, I'm going to talk about the philosophical aspect of research, particularly the science with proper methodology. So that's not something I'm going to talk a lot, but I'm going to infuse in along my talk. I want to give you a clear indication that I started to have an understanding and interest on this subject when I was working in Ralph Mitchell's lab at that time at Harvard University in America before I came to Hong Kong. But after I joined the faculty of University of Hong Kong. I start one line of research is through international collaboration with Japanese research scientists in Cambodia. And similarly, almost at the same time, I also work with the Dunhuang Institute, which is nationally with Chinese scientists. So basically, I have both lines parallel of research ongoing for the last almost 20 years. So to introduce this particular subject of cultural heritage, material is very important, and the material are an important part of our human civilization. So we started with a natural material. Human use stone and animal bone and other material they can find, including wood at the beginning. And then later on, they started to use construction material, for example, concrete, steel. And right now, we are using more and more polymeric material. And even though the negative aspect is a microplastic, but I'm focusing on macroplastic, particularly polymeric composite material. I'm going to only briefly mention about it. So the truth about human civilization and the connection to material is we are striving for durability and the strength improvement of the material. So we are going to find hard material and material we can be used for a long period of time. So that's not going to change, even though we know microplastic are a serious problem. So let's look at a little bit back about this particular issue or this concept. Humans are very different from all the other animals through the evolution of Earth. For example, we learn from one generation to the next. What does that mean is for animal, they learn from the beginning, every generation, repeating on the different environment. But for human, we transfer our knowledge from one generation to the next, and they can accumulate. And because of that, humans are very, very different. And also different in a way is we are innovative, and we are always going to creating something which can improve our quality of life, and also knowledge which can improve our ability to make new things. So initially, we have pottery, and then we have ceramic. This is the major technology improvement to have higher temperature and also pure clay material. And then by handling fire and high temperature, knowing some of the mineral, then we have refinement of bronze, metal, particularly iron, then human come into the Iron Age. And after that, because the human has been able to create more wealth, then we enjoy more and more material which are not essential for our basic living, but to improving the quality of our appreciation of life, painting, and all kinds of carving, monuments. And then we also have non-material cultural heritage, for example, musical, and that's another part. So all the previous information allow us to have a wealth of cultural heritage scattered mostly everywhere in the world. And here I'm going to use Angkor Wat as one of the examples. So I started my research at the beginning of 2000 with the Japanese research group. This is Professor Yuko Katayama, because he is the chief scientist of the Japanese team. And she invited me to join her team, and then non-stop over the last almost 20 years, obviously because of the COVID. And for the last almost 3 years, we have not been able to go back, but we are planning to be back again near the end of this year. So when we talk about Angkor Wat, especially if you talk about this to anybody, in their head, it is the Angkor Wat in this picture. This particularly monument is so important because it is on the flag of Cambodia. And this Angkor Wat in Chinese is called Xiao Wuge, actually is not the largest temple in Cambodia by the Khmer civilization, because north of Angkor Wat, it is Angkor Thom. In Chinese is Wuge Dacheng. Angkor Thom is more famous and more impressive and magnificent because Angkor Thom was a big city, and the royal palace in the city was the residence of the king. And at the same time, there are Bayon temple. There are also a lot of other functional building and also residents living in that particular compound. So here in my talk, I'm going to talk about the Angkor Wat, and also I'm going to refer to the Angkor Thom from time to time. And here is a look of Angkor Wat carving on the sandstone wall. And it's about the history, the battle of their civilization of the Khmer. And in one direction, it is 400 meters. And if you look at closely of Angkor Wat, what I want to show you here is a look at the stone to stone fitting. You cannot insert a knife into it. So what does this mean is, if you look at the quality of Angkor Wat, particularly the sandstone carving, which is very good because the fitting, the material they used was very, very good. So the construction material and the quality of construction was so good. That's allow Angkor Wat have a better condition or better appearance today. Right now is come to Angkor Thom, particularly the Bayon temple. And over here, you can see that the Bayon temple is not in similar good condition as Angkor Wat, because what you can see here is, you can see over here, the pillar of the stone was missing. And because of that, they use concrete and steel to reinforcing the support. And similarly, this part of the tower was also collapsed and missing, and here is also missing. So the reason or the comparison between the two, they were constructed at a different time of the Angkor civilization, and Angkor Wat at the peak. In that case, they were very, very wealthy. They have a lot of manpower and material. Angkor Thom, particularly for Bayon temple, was finished when the Angkor Empire was near to the collapse. And because of that, they use all kinds of material they can get to just piling up to make this Bayon temple. So material and also the technical skill are important. And this is a close look at the tower part of Angkor Thom Bayon temple. You can see from stone to stone, the gap is much, much larger than what is in Angkor Wat. And the tower at a Bayon temple have the characteristic of a Khmer smile, which attract tourists to this particular site. So to talk about technical aspect of destruction of the cultural heritage, one of the major destructing biota is the flora, which is the tree or the wood. In this case, you can see that ficus tree grow into the temple, particularly through the small gap between the stone, and then they can over the temple, they can also cause the collapse of the temple. And this particularly biological growth over the temple are so impressive. You can see it in the Hollywood movie, Indiana Jones, particularly the Tomb Raider. And in that particular case, part of the film was being shot in Cambodia and in one of the temple. And apart from the visible part of the destruction, they are invisible. One of the invisible one is temperature. For example, here, I'm not going to talk about in terms of the detail. every day from the morning till the afternoon, you can see on particular piece of the stone, temperature difference would be between 2 and 5 degrees. And if you look at for the stone facing the sun, from the stone which are in the shade, the temperature can also be between 3 and 6 degrees. And we can also look at between the biofilm and the non-biofilm stone, temperature can also be a few degree difference. And when they are rain and dry, and the temperature can be even bigger. So if you look at the temperature of, even though such a small change, but if you look at the structure of the temple, which has almost a kilometer length, then the strength, particularly internal stress put onto the stone, would be huge. And similarly, but we can also have from invisible to visible change. For example, in Angkor Wat, the materials was very impressive. The carving has been generally preserved very well, but at a certain location, you will be able to see there are damages. And this damage is very clearly due to water. So similarly here, and this is the sandstone column supporting the gallery, because this part on the left-hand part was the carving. But at the base, if you can see over here, the ground level is lower. That means allow rainwater to be trapped over here. Then the base part of the sandstone would be destroyed and destructed much more seriously. And here you can also see similar phenomena. So in this case is invisible phenomena also participate and take part in the destruction process. For this particular invisible to visible destruction, we talk about in this year's article, which has been published. So here, I'm not going to elaborate anymore. What I want to focusing on is the microbial colonization. First, microorganisms like surfaces of the material, all kind of material. They do not discriminate between material. And because of that, microorganisms, particularly when we talk about material under natural condition, which are exposed to natural condition, colonization by microorganisms is unavoidable. But microbial colonization is also important because with colonization of microorganisms and also the color is showing history. For example, if we clean all the microorganisms from the surface of any cultural heritage, visitor would have a misunderstanding. It's too clean, it's not a historical monument. So because of this, this should also be bear in mind because it's showing history. But another factor I want to emphasize at the beginning is microorganism colonization on material is not one-way street. So it's not good or bad because it could be good at the beginning and then later on going to be bad to the material. So we have to look at a different stage. So we cannot say that biocolonization is always going to be bad to the material. For this part, we have reviewed this subject almost 2 years ago, and it has been published. So again, I'm going to skip that one. And here you can see colonization of microorganisms on the surface of Bantis 3. Bantis 3 is very famous in Angkor Wat monument because it was being constructed to memorize the mother of the king. But it was miniaturized because the temple is very small. The construction material is slightly different. You can see the color is red, but the carving is very delicate and meticulous. And because of that, conservation, protection of this temple is one of the high priority in among all of the monuments in Angkor Wat. And if you look at one section of Bayon temple, this picture is illustrating, you can see that if this part has no serious constant water intrusion, that means water going to be impact this area, but in the adjacent area, they see the whole section of the carving. But in this part, the back have constantly water, which would be sipped into this section. So you can see this part is completely covered constantly by microorganisms, and then it's blackened and also deterioration. So because of this, what I want to emphasize over here is if we want to deal with the management of Cambodia, Angkor Wat, and other monuments, water is one of the key issues before we talk about the biology. Another issue we need to address, I'm not going to talk about too much in detail, is the sampling technique, because cultural heritage are precious, unique. You cannot use extensive scraping or destructive approach. So in this case, we use one particular technique here is being shown, use an adhesive method to take the surface of the microorganism and then to do further analysis. By using this particular technique, we can take what is on the surface of the sandstone material and then take the material back to the laboratory. What here I want to emphasize is you can take a sample repeatedly at this particular site, one after another, as many times as you want. By doing it this way, this is what being shown through our effort. At one particular location, we took almost 40 sampling at this particular site. Here is illustrated. This is the first one. That means on the very top, and then subsequently number 9, 17, 28, and 36. Through analysis of the sample at a different layer, what we can show is the surface layer is dominated by cyano microorganism. That means photosynthetic microorganism. In the deep layer, in the bottom part, then they are oxygen sensitive or facultative microorganism in the lower layer. So by doing it this way, you can show the spatial distribution of different microorganisms. Otherwise, it's not going to be allowed to be illustrated. So this technique, sampling multiple at one location, has this advantage, and this is what I illustrate here. And since we talk about the sampling technique, I want to give you a briefly touch on the history. Initially on this topic, we were using isolation and purification to identify the microorganism from cultural heritage. So this technique is used much less today. And currently, most people use genomic approach, particularly using high throughput sequencing to get the community composition of the microorganism. And this approach is simple and much efficient. But what I want to emphasize here is you can get the information of a microbial community as what we did at Tower Gallery number 42 at Cambodia. But we do not have the information of the biochemical reaction and also the active microbial group in this community, because it is genomic DNA, it has no relation to the metabolic active microorganism in this case. So similarly, we can also sampling different section and different color on the carving of the sandstone from biosample. We know the matching between the color of the different microorganisms. Again, there is no mechanism or further information on what is the biochemical reaction and how microorganisms contributing to the different color. We only know the microorganisms are the dominant one on the surface. So because of this, high throughput sequencing, metagenomic have to be used very cautiously and have to be used with a specific question. This is in my opinion, and we also published this information last year and before last year. So we want to emphasize the active microorganism in this case. And for this part, I have a new article, which would be published soon, is I talk about the microbiology in terms of the chemistry and the physics. Without a good understanding of the bottom part too, then the microbiology on the top cannot be a solid science. This is going to be elaborated later on. Because of the time, I have to move on slightly faster enough. But I want to give you one particular information here is at Angkor Wat, we can do a simple experiment. And we look at this particular column supporting the gallery, because the gallery is on the back. This is the Professor Yuko Katayama. She was sampling the top part of the column. We take a sample, this is from A. We take another sample between the destruction can be visible, because above it, there is no visible destruction. Below it, there are visible destruction. This is the B. And then from the bottom, destruction has already resulted in loss of the sandstone material. If we took these three samples and to the back to the lab to incubate in a sulfur oxidizing media, then you can see that the lower bottom part of the sample within 5 days can result in a pH 1.2 unit decrease, a significant decrease of the pH within only 5 days. And after continual incubation for more than 2 months, 72 days, the intermediate part, transitional part, and also top part only result in no pH decrease or minimum amount of a pH decrease. So what does this mean is in the bottom part of the sandstone, we have a sample, there were already a lot of sulfur oxidizing microorganisms in that sample. So at Angkor Wat, we isolate the bacteria, microbacteria called microbacteria, which can oxidizing sulfur. You can see that when it's growing with sulfur inside, inoculated, the culture media is turbid. If we look at ATP with adding of sulfur, there are more ATP in the culture media. Without sulfur, the ATP are much less. And in addition to that, with sulfur added, the pH would have a significant drop, and without sulfur added, the pH is relatively stable. And similarly, not only bacteria, fungus is also contributing sulfur oxidation in this case. And by using a petri dish culture media, inoculate the fungus at the center, you can see in 7 days, there is a clearing zone. That means the fungus has been growing, sulfur particle has been utilized, and then if we analyze the 2 sections, the non-clear and the clear section, we can see that there is a high concentration of sulfur sulfate and also sulfate being produced. That means elemental sulfur has been oxidized by the sulfur oxidizing fungus in this case. So for this part, the beginning part, I want to give everyone a common understanding that microorganism, bacteria, fungus, they like sandstone. And the microbial colonization on the sandstone can be destructive, but is not clearly always going to be destructive from the beginning. And what we need to do is we need to focusing on the biochemical reaction and the specific mechanism by microorganism, which are contributing to the destruction to allow us to understanding the process. So sulfur oxidizing by bacteria, by fungi are bad, and they are the agent which are causing deterioration of sandstone. Again, it has to be in the presence of water and understanding of the water cycle. So now I want to give you one specific information on microbial contribution to the destruction of sandstone and what is information we have generated. For science, the purpose for scientific research is we want to make something which is less known or unknown to something known with clarity. Clarity means we want to have a fundamental, solid ground explanation on what we know after our effort. So this is what is in science. And in science, we need to use and implement proper scientific method. And with the proper scientific method, we would be able to reveal what is not clear, which is not known, to something which is clear to us now. So this is the method we are using. Let's think about nitrogen transformation. I'm not going to go through all the different processes, but let's go back to Bayon Temple again. And here is an entrance to Angkor Thom from the south gate. And Bayon Temple is the most important one in Angkor Thom. And through our early days of research, we had found there is a higher concentration of nitrate in the sandstone. And you can see here at this particular gallery, this is tower number 26. This is the carving stone. Nitrate concentration are several hundred times higher than ammonia and the nitrite. To find lower concentration of nitrite is not unique, because the nitrite is always going to be in relatively lower concentration because it's toxic and it's a transitional byproduct. But ammonia concentration most of the time are comparable to nitrate, but in this case, these two are not in proportion and they are out of proportion. So this attracts our attention. So what we did, particularly by our group, we sample at the Bayon Temple. Bayon Temple is a tower temple. We sample at the ground level, and this is the base of the construction. You can see the destructed part of the Bayon Temple, but we can collecting biofilm from the sandstone. Then we go to the gallery section, which is one level higher, and the gallery I have shown you before with destruction and water seepage. We can sampling over here. Then we go to the central tower because this part is not accessible to tourists, and we can also use this one as a reference point. So through our analysis of ground, gallery, and also the tower, we found that there is a general trend of ammonia-oxidizing archaea are much higher in abundance than ammonia-oxidizing bacteria. So in this case, is ammonia is being oxidized to nitrate by ammonia-oxidizing archaea, not bacteria, because they are outnumber the ammonia-oxidizing bacteria. Since our first set of results was based on genomic, as I mentioned, genomic does not indicating activity. So in our subsequent visit, in the next year and after next year, we sample 3 temples. In addition to Bayon Temple, we including what are severe and also non-chrome. And by doing this way, you can see that the general information still hold. In another way is the AOA, the black bar are still outnumber the red one. So ammonia oxidizing archaea are still outnumber when we are analyzing the transcriptome PCR result. So the activity of AOA are still outnumber of AOB. Further to our PCR analysis, what we did is we carry out a simulation. We carry bottled water onto the top of the gallery section, and the water we pouring onto the top of the gallery was equivalent to 4 millimeter precipitation. In another way, rainfall. Then we collect the rain, not simulating rainwater, carry out into the lab in Cambodia, and then to do the quick on-site analysis. In this case, we can detect higher concentration of nitrate, sulfate, and small amount of ammonia and nitrite. And in this case, the simulation support what we have observed on Angkor Wat and also the different temple. By observing higher concentration of nitrate, it only allow us to say ammonia oxidation are very active producing nitrate. But we do not know why removal of nitrate was not active. So because of that, what we did was we used nitrogen N15 stable isotope to monitoring the nitrate removal to nitrogen gas. Because nitrogen gas produced can be from at least two different processes. One is denitrification. This is something we know a lot. And another process is the Anammox process, because denitrification uses the common nitrate. So another way is they use nitrate as a substrate to produce the nitrogen gas. If they use labeled nitrate, then two labeled nitrate producing labeled nitrogen gas. If the nitrate was not labeled, then they would produce the nitrogen gas, which is non-labeled. But for Anammox, we can put labeled ammonia or labeled nitrite, or both of them are labeled. By doing it this way, we will be able to have the paired nitrogen from labeled and non-labeled as nitrogen gas. Or we can have both of them labeled, but the gas is labeled. By doing it this way, by monitoring the gas produced, we can trace back to which process was contributing to the nitrogen gas produced. And in this case, we carry out a large number of different temples, as many as we could. But the result is clear because Anammox process was not active. You can see it throughout of the temple we have tested. Denitrification was detectable, but the rate can be variable to almost 100 times of difference. And at the same time, because we have a control in this particular case from a pond nearby to the Bayon temple, you can see that in the pond, Anammox and denitrification was almost 50% to 50%. So the reference point tells us the activity of Anammox and the denitrification in our controlling sample, but the activity was not that high in all the temple, and they were much, much less. For Anammox, they were almost non-detectable. This result tells us the nitrate produced cannot be removed very effectively, which result in accumulation of nitrate. Since nitrate are being accumulated, we know the initial AOA was very active and the removal was not active. This still does not explain fully about why nitrate can be accumulated. But last year and this year, our results have provided new information about why nitrate are being accumulated. This is from a PhD degree thesis. What Dinghua has been found in this particular case is on Angkor Wat temple and also other temple in Cambodia and elsewhere. They are Comammox process. This is a process which can oxidize ammonia through one step reaction to nitrate. This means there are other processes which are contributing to the nitrate production. At the same time, he also found another more interesting, important process, which is nitrate produced are being dissimilatory reduced to ammonia. And by finding this process, I think it's more important because this allows us to understanding how can nitrate concentration can be maintained high and ammonia oxidation can also be carried out through time because the accumulated nitrate can be dissimilatory reduced to ammonia, which is a substrate to AOA AOB, and also Comammox to produce nitrate. But the accumulated nitrate, small amount can be denitrified, but they can also be converted to the substrate to maintaining this simple cycle to allow nitrogen to not be lost and also to be on the surface of the stone. So currently what we are discussed and also try to formulate our story about nitrogen transformation on the stone cultural heritage is ALA, AOB are contributing to nitrate production together with the Comammox, but the nitrate produced are small amount converted to nitrogen gas, and nitrate can be reduced to ammonia, which would be a substrate for the oxidation process taking place. What we should not forget in this whole process is ammonia oxidation is not for nitrogen transformation alone, because ammonia oxidation is a lethal and also trophical process. And because of that, ammonia oxidation are also incorporating CO2 onto the sandstone and allow the surface of the sandstone to be enriched with organic carbon. So this nitrogen transformation changing the substrate on the surface of sandstone and then the microbial ecology. So that's something I want to highlight in this particular part of the talk. And this finding of nitrogen transformation, nitrogen cycle in Cambodia is not in Cambodia alone, because we also analyze other temples in Cambodia and also data from other cultural heritage on other sites globally. And similarly, recently, our collaboration with Dunhuang Academy has also produced a similar result on the nitrogen cycle with Meijishan and also Beishiku. So the phenomena we have observed is more widespread and also more common. And I'm sure other people, when they are looking at a similar process, they will also be able to confirm what we have found. And because of the time is restricted, so I would briefly go into this part. Microbial ecology and microorganisms are important. That's not the only information to explain how stones are being destroyed. Stone destruction involves water cycle. Is also involving the drying process, which is a recrystallization of the soluble salt. And for this part, the detailed information are being discussed in this book. If anyone interested in more detail, you can read, because this book is very good. It's an open access book, so you can search it on the internet and also through ResearchGate, and you can read it. The main information I want to emphasize in this part is cultural heritage research, particularly in terms of microbiology, need knowledge, need technique, which are chemistry, biochemistry, mineralogy, and also material science. So we cannot talk about microbiology based on microbiology to explaining cultural heritage, destruction and protection and management. We need to know the different subsets. Without them, we cannot really do anything effectively. So because of this, what we have done is, you can see a sandstone material, sandstone sample from Angkor Wat, and without exposure, this is the after exposure, there are products being formed. And you can see in this case is the major difference in the elemental composition is calcium as an element has been decreased over exposure, but obviously over exposure, nitrogen concentration has also been enriched. But for calcium being depleted, we have been focusing on mineral. This is by SLD analysis. And we have found that calcite, which is a calcium carbonate, has been decreased over time of exposure. Calcium carbonate as a calcite is a mineral, but do not take as an insoluble mineral alone, because when calcium carbonate is soluble, then it is a bioavailable carbon dioxide to microorganisms. So because of that, dissolution of calcite and calcium carbonate, which is the inorganic carbon, to ammonia-oxidizing archaea, Comammox, and which would allow them to oxidize ammonia on cultural heritage, and then this would allow the dissolution of calcium carbonate, which can enlarge the pore in sandstone, which can encourage the biofilm formation, microbial growth, and through the whole theory of reaction and also synergism of microbiology, biochemical reaction, mineralogy, then there would be a rich microbial community. So in my talk, what I have emphasized is material are important part of our civilization. We are going to improve the quality, durability of the material. That's our goal. But when we look at the natural material, particularly their deterioration, we need to look at water cycle, microorganism, and also the salt, particularly the salt in terms of soluble, insoluble movement of the salt, and also recrystallization of the salt. So the research require methodology innovation. The non-destructive sampling technique is needed, and we need to look for better technique to reveal the microbial community, how microorganisms and what is their distribution, particularly spatially. And by looking at that, we can look at the microorganism on cultural heritage and reveal microorganism on the surface. And by doing it that way, we know sulfur oxidizing bacteria, fungi, how do they carry out sulfur oxidation. And through looking at the nitrogen cycle, we know AOA, AOB, Comammox contributing to ammonia oxidation. We also know through further analysis that Anammox is not going to be an important part, but the denitrification contributes to some part to the removal of nitrate. But at the same time, DNRA is more important because it's allowed to maintain the nitrate on the surface of the sandstone and allow cyclic reaction of ammonia oxidation. All the information of sulfur and nitrogen cycle is reinforcing my argument. Chemistry, biochemistry are important to the microbiological understanding of cultural heritage destruction. And because of that, what I'm arguing here is in terms of the methodology, in terms of the philosophical question is in terms of the research. Our research paradigm is shifting or is needed to be shifted because we need to look at microbial biota, the microbiome on the surface of the sandstone, not simply their composition, not simply about the biochemical can be identified by the CaC analysis, but by the specific biochemical reaction, the material, the gene expression. By doing it that way, we can reinforcing our analysis and also of our knowledge and information. With that, I would finish up with my talk. I want to thank the Japanese Trust Fund because they provide the financial support for our travel and the logistic over almost the last 20 years. And also Apsara Authority of the Cambodia Kingdom, because they allow us the research to be carried out in Cambodia at the different temple. At the same time, I also received research grant from Hong Kong government and for the research carried out on Angkor Wat before I left, and also Dunhuang Academy for the last almost 20 years on different tempo and a different part of the research. And obviously, I also want to thank the students, postdoc, and they were involved in different part of the project, and I couldn't name one by one because of the time. And this is our latest picture of our research in Cambodia. And I hope we can go back again to continue our work either at the end of this year or next year. I want to thank you again, and I just want to finish my talk here. Thank you very much.